then only it can show action. So basically the action will depend on the drug receptor interaction. So if the drug receptor interaction is there, then only the action will be there. Okay. So we see that the magnitude of the response is proportional to the number of receptors complexes, or simply how many receptors are occupied by the drug molecules. So it's simple way, in similar fashion of the reactions kinetics, we have seen that drug plus receptor, that gives a drug receptor complex. Okay. And that next constant gives you response. Okay. So and there is a dissociation also in between. Okay. So if the drug receptor, whenever it is dissociated, then the complex is less. Okay. So drug plus receptor causing, or that is association constant, we will call it as a K1. K1 is association constant. K2 is a dissociation constant, that is the drug is dissociated from the receptor, and K3 is responsible for the response. Okay. So these are the rate constant, because as I told you, it will depend on the proportionality. That more the number of receptor are occupied by the drug, more will be the drug receptor complex. If more is the drug receptor complex, so it is proportional to the response. If more is the drug receptor complex, more will be the response. So this is very simple that way. So the rate at which the drug molecule combine with the receptor, that is what we call it as association constant. The rate at which the drug receptor complex dissociate, that is K2. Okay. And the rate at which the response is generated following a drug receptor interaction is given by K3. So these constants are used to define some of the basic concepts which are related to drug receptor interactions. Okay. And these concepts are useful in quantification. Whenever the quantifications are required or possible, so at that time these quantifications are useful. We know about two things. Okay. That for every drug must have two important properties, that is ability to bind with the receptor and ability to start the action or generate the action. And we call these two things as affinity and intrinsic activity. This is we have seen multiple times. Okay. So this is when there, let us see what will happen and how other things are defined on that basis. So next thing is that is called as a potency and efficacy. Potency is always there, or it is always depending on the ability of drug to bind with the receptor. That means what? It is depending on what? Ability to bind with the receptor means yes. Ability to bind with the receptor means what kind of property? Very good. It is affinity. Okay. So the drug which are having more affinity, they will have more potency. Okay. So let us see what it means. The drug is said to be potent when it possesses high intrinsic activity at low unit weight doses or at lower concentration. We have discussed many times, there are drugs which are active or their dose is in micrograms, 10 micrograms. You can understand 10 microgram means, okay. So it is 10 microgram means almost 100 part of 1 upon 100 part of milligram. Okay. So there are drugs which are active in microgram, there are drugs which are active in milligram, there are drugs which need the dose of 100 milligram, there are doses which need up to 500 milligram. So accordingly, that means what? Some are potent, those which require higher dose, they are less potent. Okay. Yes, dose is not only depending upon the potency also, but just to imagine, just for, because we have seen that pharmacokinetic parameters are also important. We have seen that for any drug which is having 80% protein binding or less absorption or first pass mechanism, in that you have to give a higher dose, so that to compensate the losses in between. Okay. So knowledge of drugs potency is important for finding out the appropriate dosage level to be administered. Then second thing is efficacy. It refers to maximal or peak response that can produce by a drug, and it is important determinant in the drug selection process. Again, let us go back for our original example. For this, we have given the example of what? Car and driver. Okay. Now let us go ahead with the two new definition which we had. What are these two definition? Are potency and efficacy. Now car are same. Okay. And in this car again persons are coming. When the persons are coming, we know there are two kind of person can be there. Those who can drive, those or in forward direction, and those who cannot drive or drive in backward direction. Those who can drive in forward direction, we call them as agonists. Those who drive in backward direction, we call them as inverse agonist. Those who cannot drive but occupy the car and because or car driver seat, and because of that they do not allow actual driver to drive the car, and because of that they causes delay in the driving, we call them as antagonists. Perfect. Okay. Now let us make this. There are various drivers. Okay. Out of that, there are some drivers who are very fast. As soon as it is told, they immediately move to car very fastly. They immediately enter the car without delay. Okay. And without delay, when they enter the car and drive the car. Okay. So the speed with which they reach to the driving seat, and what with ease what they open the driver's seat door, that is what we call as the affinity. Those who can do it fast, these are the having higher affinity. Okay. Then what about the efficacy? Now all the drivers, they are able to do it. Driver 1, driver 2, driver 3, driver 4. Okay. Now those who entered fast, they can start car fast. But there are the person, so initially they will be ahead. Initially they will be going ahead because they are faster. They immediately reach to the driver's seat and they started the car, and they have started car in a forward direction. There are now in the driver there are two type of again people. There are the people who can drive it faster speed, and they can drive, they are very skilled driver. There are other drivers which are driving at a slower speed. They cannot drive at very fast speeds. Okay. Both can reach to destination. Okay. But number of, suppose as I told you, from our college we have to drop the people to railway station. Who will able to drop more people? Those who can open the car door fast, those who can drive fast. Okay. They will be doing more job. Those who can open car door slowly, they are taking time in there. Okay. Then it will take time. Now depending on the further actions, those who are having lesser affinity and lesser efficacy, they will be having slower activity, or they can just behave similar to that of partial agonist. Okay. While those who are having higher potency and higher efficacy, they can do more job. This much you understood, potency and efficacy. Potency is depending on binding abilities. Okay. While the efficacy is always depending on the intrinsic activity. How much intrinsic activity drug possesses. So intrinsic activity means ability to drive. Okay. And potency is if they can do it faster, more receptor can occupy it. Okay. So this is the affinity, this is the definitions, or this is the explanation about What? Potency and efficacy. These terms are very, very commonly used. Always, wherever till you are there in this field, the terms like agonist, antagonist, potency, efficacy, they will be very, very commonly used, and you should be aware about it. So always keep in mind, potency doesn't mean that the drug is very powerful. Efficacy means the drug is powerful. Potency means it has ability to bind more number of receptor at a given time, because it has got more affinity. So simultaneously it can bind with more receptor because it has got more affinity. Okay. While the intrinsic affinity means once it binds the receptor, it can cause, suppose you have studied, you know about the second messenger. So drug which is having lesser intrinsic activity will able to generate lesser second messenger or lesser third messenger, or if it is a cell membrane type of transporter receptor, will able to open it in a lesser fraction, while those which are having higher intrinsic activity, they will open it in more way, so that more ion can be transported, or they will able to generate more second messenger. This is clear. What is efficacy? So efficacy means they will able to do the job more, but potency means what will happen? Maybe they are doing it slowly, but they can occupy more receptor because they have more affinity with the receptor. So because of more affinity, they will bind more and more, and because of more binding, maybe initially they can able to give more response, because or faster response, because they have more affinity. Immediately as soon as they come in contact with that, they immediately bind to the receptor. Okay. So the drug which is having higher potency and higher efficacy, they are the most powerful drug, or they will require the most less dose. Okay. Lesser and lesser dose if the potency is higher and efficacy is higher, and those which are having lesser potency, they will require higher dose. Those which are having less efficacy also, usually we can call them as a partial agonist, because they are not that powerful as the agonist. So this is how potency and efficacy are important. Okay. This much is clear. This is understood. So any response, so the drug response will be depending on the drug receptor interaction. We have seen many times, I told you many times, that higher is the drug receptor interaction, higher will be the activity, higher will be the response. When the higher response will be there, but the drug must have both these things, higher potency and higher efficacy. So more will be response. So currently 3 theories are there to explain the nature of drug receptor interaction and their relationship to the response one. That one is, first is receptor occupation theories, second one is called as a rate theory, and third one is called as a theories that involve the drug induced protein changes at the receptor size. Okay. So these are 3 important theories which govern or which indicates how the drug will be showing response. Okay. And some of these can be used, or these theories can be used for quantification of the response. So first theory, what it says, it postulates, the word comes as a postulate, that means it is not very sure, it is what, it is the imaginations of the scientists, or it is the belief of the scientists on their observation, because they don't have anything which, by which they can prove it correctly, but whatsoever observation they have got, they feel that, they expect that, therefore there is a word that comes as a postulate. Postulation means hypothesis. Okay. And when it is confirmed by various studies, then it is, instead of it has observed or it is found, this kind of word will come. Now postulate word will not come. You understood the difference between postulate and assumption? Yes, it is assumption. So this theory postulates that the magnitude of the response produced by the drug is directly proportional to the number of receptors which are occupied. So simple, more of the receptors are occupied, more will be the drug effect. So maximum drug effect will occur when all the receptors are occupied. So this type of thing is called as a receptor occupation theory. Okay. The second theory is there, that is called as a rate theory, and that postulate that we have seen that it is a dynamic. It is not that because the car model is that way easy to explain, but it is not that easy. Why? Because actually in receptor side it is not like that the person is sitting for a long time. There is a constant association and dissociation is going on. Okay. So if the rate of association is higher, then the effect will be higher. So every time it associate, elicit response gets dissociated, get associated with the response, it starts the response, maybe transporter 1 or ion transport, or maybe second messenger, whatsoever. Okay, it binds, it starts response, it is dissociated, binds means associated. So this is continuously going on. So this is a dynamic reaction. It is not as easy as we said in a driver or car. That was for easier understanding in initial phases. But now as we go ahead, we should understand that. So rate theory postulate that the intrinsic activity of the drug is related to the rate of complex formation, that is K1. Okay. If the K1 is higher, then more association will be there. Okay. And the rate of complex breakdown, which are in turn causes the association and dissociation of the drug with the receptor. So every time association, dissociation will be there, every time the action will be there. So agonists, they have high rate of association and dissociation, and again they possess both intrinsic activity also, okay, or efficacy also, and therefore the turnover number which is deciding the magnitude of the response. While antagonists, they produce the stable complex. It is not sure, we shall see different kind of antagonists, but complex with the receptor, and that does not allow the agonist to bind, and therefore it slows down the association of agonist and the receptor. The association between agonist and receptor, that is reduced because of antagonist, and therefore if the association is reduced, then the drug receptor complex formation is reduced, and if the drug receptor complex formation is received, the response is reduced. Okay. So this is how this rate theory work. And the next theory is called as the theory that involve the drug induced protein changes. Okay. And that makes that there are agonists, they causes action by inducing some temporary structural changes in the protein part of the receptor, and each structural alteration modify the permeability of cell membrane, and it produces the response. And antagonists produce structural changes to prevent the formation of agonist receptor complex. I have explained you this with the example of benzodiazepine, if you remember. Benzodiazepine is not acting exactly on GABA receptor, it acts at somewhere else, and when it binds with the GABA receptor, that modifies, we call it as allosteric site. Allosteric means which is not actually site, which is something different site, which causes such a modification that there are changes in the receptor. So because of this kind of changes, the GABA mediated actions, they are improving manyfold. Okay. And thus benzodiazepines are working. So benzodiazepine structures, like structures are not present in the body, but GABA is there. So GABAergic drug act on GABA receptor, but benzodiazepine acts on a benzodiazepine receptor, and they alter the channel structure. Okay. That chloride anion channel structure they alter, and because of that alteration, they allow more chloride anion, making the inner side more negative. If it becomes more negative, if you remember last year impulse conduction, then we call it as a IPSP, inhibitory post synaptic potential. So this is what the benzodiazepines are causing. So like that, there are many such things that because of that alteration of protein change and the receptor side, this is happening. Now, why we are studying this receptor concept, or what is the importance of this receptor concept? So, we understand the roles and function of the receptor because most drugs interact with the receptor. Okay, and the drugs which bind to receptor, they can be of both type. They can be agonist or they can be antagonist. Very important thing is we cannot have antagonist for non-receptor mechanism, isn't it? Simply because we have seen non-receptor, there was some alkaline agents or bases, they were taking care of acid. There is no receptor. We are not antagonizing any receptor. So no. Okay, Pallavi, now it's clear. So there is no more. All others are having clear. Okay. Okay, Pallavi, check then. Most probably your net connection has a problem. Okay. Now the thing is, so therefore we need to study, and therefore receptor they determine the quantitative relationship between dose of the drug and pharmacological, and to that we call as a dose response. Okay, more is the dose, more is the response, like that. But that response will be depending on these both abilities. What these are the abilities, what is the potency and what is the efficacy of the drug. So then we shall go for later some graph. After a few slides we may go for some of the graphs which explains this drug response concept. Okay, so these graphs are there that will help us to understand all the things about agonist, antagonist, potency, efficacy, and we shall see them later. But by that time you should be very clear in these four. You should make it clear in your mind. If somebody asks you what is agonist, what is antagonist, what is partial agonist, what is inverse agonist, okay, then what do you mean by potency, what do you mean by efficacy, okay, then this, if it is clear to you, it should be almost just like now somebody asks you about table of 2, table of 4, or some simple spelling, that or this kind of spelling, and you can write it. Similarly, that much ease you should have in understanding this basic concepts of pharmacology. This much is clear. So we have seen what is agonist, okay, we have seen what is antagonist, we have seen what is partial agonist, okay. I told you they are very important. Now if somebody asks you for 5 marks, there are many points you can write. You can write about affinity, you can write about the intrinsic activity, you can write about the affinity, you can write about the affinity, intrinsic activity, you can write about potency, you can write about the efficacy, okay. So what they are and how they are causing, what are the principles, this is what agonist you can write. You can write anything about agonist. Similarly, antagonist, you know about antagonist now, we have seen it in detail. Now you have seen the partial agonist, you have seen inverse agonist, okay. There is particularly only one example is there, that is of beta carbolin, which is bind to GABA receptor, and it causes stimulation, anxiety, and increased muscle tone and convulsion, because that causes changes so that the GABA cannot act and make the chloride transport. So that chloride which naturally was going will not able to go. Now there can be some agents which are mixed agonist and antagonist. Particularly this is happening when there are multiple type of receptor. So some of the agents, they act as agonist on one kind of receptor and antagonist on other kind of receptor. This is particularly more important when we study opioid analgesics. In that we study that there are drugs like pentazocine and nalorphine. They causes severe psychotomimetic effects which are not reversible with naloxone, okay. and they means there are let me tell you, there are variety of receptor like mu, kappa, delta, okay, and these receptors are there out of sigma, and out of that when they act on one receptor and they do not act on other receptor, or they causes inhibition on other receptor, then we call them as a mixed agonist, mixed antagonist, okay. So they are rarely found in other cases, but yes, they are available. They are in opioid type of drugs which are used as a painkiller or morphine like drugs, okay. Apart from that, there are many other types of antagonist which I shall tell you after going through some of other topics, okay. There are antagonists we can classify on many other bases. So far we have understood what, that there are agents which are acting as agonist, there are agents which are acting as antagonist. We have seen that receptor can be of variety of kind. We have seen one more thing is the in rate theory we have seen that either drugs can act on the receptor itself, or it can act on nearby proteins, and because of acting on nearby protein, it alters the receptor in such a way, or it alters the whatsoever it may be, whether it is a transporter protein or whether it is a G protein type of protein, but it alters in such a way that it causes the changes, and because of these changes it causes the action, okay. Now similar way we can have antagonist which can act either directly at the receptor site or the site of agonist where the agonist is binding. Example where you have seen last year autonomous nervous system or hormones. So we can have an antagonist which can exactly acting on the site, like we have got various kind of receptors in adrenaline, noradrenaline. So we have got alpha receptor, beta receptor. In beta we have got beta 1 receptor, beta 2 receptor, beta 3 receptor. So like that variety of receptor we have got. So we can have drug, suppose drug which is acting on beta receptor or beta 1 receptor, then we can have agent some agonist which is directly acting on beta 1 receptor, or there can be some antagonist which is directly acting on beta 1 receptor, we call it as a atenolol, okay. So like that this is a very direct one, but there can be some indirect action also, as we have seen in benzodiazepine and in other cases. Similarly we can see the such kind of mechanism in antagonist, okay, and what kind of binding they are making. On that basis we can have a variety of combinations, and these combinations we can see in the next slides. Further in between we can go for the what is the mechanism or what are the mechanism of signal transduction. To what we call as a signal transduction is that somewhere some part of the body gives the sign, okay, and that is in the form of some chemical release. If you have seen autonomous nervous system last year, we have said that it is a fight and flight mechanism. So that is what signal to fight against some problem, okay, and because of that you might have studied that it causes this flight and fight mechanism that causes activation of sympathetic nervous system. That means what? So first thing is the brain senses that there is something dangerous situation, and we either the body has to fight with it or body has to run away from it, to what we call it as a fight and flight. So if the body has to do anything of this, then body has to be prepared, and for that it gives signals to the body in form of adrenaline, noradrenaline. So adrenaline, noradrenaline will act at one or other places. So when it acts at one or other places, that we know that it can act on blood vessel, it can act on heart, it can act on liver, it can act on various places through variety of receptor, alpha, beta, again alpha 1, alpha 2, beta 1, beta 2. There are many receptors in ANS. Now it is acting on that receptor. Now suppose it is acting on beta receptor. So because of that, acting on the beta receptor or beta 2 receptor, it can cause the initiation of the formation or starting the second messenger. Okay, so this whole process from first release or from binding to the receptor to actually initiation of the effector activity, we call it as a signal transduction. Okay, so this signal transduction, how it takes place? Usually the most powerful or most important structure in that, there are variety of receptor. One popular group of the receptor, they are called as a G protein coupled receptor, or very shortly they are called as a GPCRs, G protein coupled receptors. Okay, they control many aspects of the cell function by acting on variety of different signal transduction mechanism. There is a link between membrane receptor and first stage of the signal transduction cascade, and it is established through G proteins. There are a variety of G proteins, we shall see them. Okay, then G protein represent the level of middle management in the organizational hierarchy. They intervene between the receptors and the effector enzyme or ion channels. They are called as a G protein because their interaction is there with the guanine nucleotides. So guanine nucleotides, they play important role in function of this. Therefore, these guanine nucleotides are GTP and GDP. They are also called as a metabotropic receptors because they alter the metabolism. Okay, and therefore they are also called as a metabotropic receptor. All the monomer are there, and they monomers of these G proteins, they form 7 membrane spanning segment, and that all 7 membrane, they are the across the cell membrane. So it is also called as a 7 transmembrane segment. So all these monomers are there, and it is very peculiar structure that wherever G protein is there, it is always a 7 membrane. Okay, or 7 membrane or 7 loops are formed because of that. All the intracellular loops are larger than the others one, and they are interact with the G protein. G protein is the membrane protein which comprises subunits like alpha, beta, gamma. Okay, the alpha subunit is having GTPase activity, just like we have seen ATPases, ADPases. Okay, similarly it is a GTPase activity, and there are 3 main classes of G protein: GS, that is stimulatory action; GI is inhibitory action; and GQ means a miscellaneous type of action. Okay, so we shall see these all G proteins and the signal cascades we call. Cascade means हिंदी में जिसको झरना बोलते हैं, मराठी में दबदबा, है ना. जैसे पानी एक जगह गिरने के बाद वो नेक्स्ट 2 rocks पे गिरता है, after 2 rocks it goes on 4 rocks or like that. So one after another, what happens? This is called as a cascade of reaction. So this kind of cascade of reactions we can see with the G protein, and we shall see all other one where there is a signal transduction takes place. So signal transduction plays very important role. Signal transduction is a very typical features of all the receptor mediated drug action, and therefore anything goes wrong during signal transduction. अब तुमको याद आता होगा कि मैंने बहुत सारी बार फ़ैन का एग्जांपल दिया था कि अगर फ़ैन में प्रॉब्लम आता है तो क्या होता है. तो फ़ैन में इतने सारे कंपोनेंट होते हैं, तो फैन में यहाँ से अपने करंट छोड़ने के बाद फैन में पहुँचने के बाद भी देर आर सो मेनी कंपोनेंट्स आर देयर, एंड इवन वन सिंगल कंपोनेंट गोज रॉन्ग, स्टिल द फंक्शन इज नॉट प्रॉपर. सेम थिंग इज इन देयर इन जी प्रोटीन कपल रिसेप्टर. अगर मान लो जी प्रोटीन बाइंड हुआ, उसके बाद एडेनिल साइक्लेज हुआ, एडेनिल साइक्लेज से साइक्लिक एएमपी फॉर्म हो रहा है, अगर वो ठीक से फॉर्म नहीं हुआ, सफिशिएंट क्वांटिटी में फॉर्म नहीं हुआ, तो एक्शन कम हो जाएगी. अगर किसी में ज़्यादा फॉर्म हो रहा है तो ज़्यादा हो जाएगी. देन दिस एडेनिल साइक्लेज इज मेटाबोलाइज बाय द एंजाइम कॉल्ड एज फास्फोडाइस्टरेज. किसी में ज़्यादा है, किसी में कम है, तो वापस एडेनिल साइक्लेज का मेटाबॉलिज्म कम या ज़्यादा होगा. And accordingly we can have variety of places where we can concentrate on. This is clear. Now we are getting complex and complex. I am therefore going slowly. Understood this? G protein coupled receptors. Cascade, you understood word? Okay. Signal transduction word you understood? So you note down, today we learn few important words. We learn about signal transduction. We learn about the terms about affinity, intrinsic. Yes, we learn two important things about signal transduction is one thing, and second thing we learn about what? We learn about various concepts in agonist antagonist. Particularly we learn various theories of drug action, and we learn about what? The definitions, like what is the potency and what is the efficacy. So many terms we are learning. You can make the note of it. You can define all these terms. Try to make a comprehensive note, so that if somebody asks it for 1 mark, you can just define what potency is, what efficacy is, and if somebody asks for more mark, you can add the things into it. Okay. So we shall study about it in the next one. What kind of signal transductions are there? I hope this much is clear.